So thank you everyone for coming. And welcome to the Lower Columbia chapter of the Ice Age Floods Institute. And tonight we are having, our speaker is Andrew Boehm. He is a research archaeologist with the Museum of Natural and Cultural History at the University of Oregon in Eugene. Tonight, he will be speaking on new Mastodon species, the current state of research. So I'm going to turn it over to you, Andrew. Thank you so much for coming.
Well, thank you so much.
There we go.
There, I hit the right button. Okay. I don't get to do this very often. So, yes, first of all, thank you so much, everyone, for attending tonight and coming out. I had no idea there would be so many. So this is wonderful. So as Sylvia said, my name is Andrew Boehm from the Museum of Natural and Cultural History down at the University of Oregon. And tonight we're going to talk about basically mastodons in general, and then specifically about the mastodon in Tualatin and kind of how it fits into the state of research that's going on right now within mastodons. But keep in mind, I am an archaeologist. I'm not a paleontologist or geneticist, so I'm going to touch on some of those things. But if you ask some questions, I will admit when I don't maybe know as much as I could if I was more specialized in one of those fields, but just to give you a heads up. So first of all, I really have to thank this wonderful group of people that have helped all along the way. No project is ever done by itself, and this is especially true in this case. First of all, I want to thank Yvonne Addington, who has basically just been the key figure in making all of this happen. I was able to reach out to her, and she was able to connect me with the Historical Center and basically everyone else, and really got this ball rolling. And then was able to help me better understand the history of not only. And Ed Davis has helped a lot getting access to the collections. He's our paleontologist down here. And I also want to thank our museum here for providing me not only with a job, which is always wonderful, but the time to go out and do these really fun projects and working in the area. So to kind of kick off, I wasn't sure how much everyone knew about mastodons in general and kind of proboscideans. But when we think about proboscideans and big elephants, we usually think of mammoths, particularly woolly mammoths, because they've been in TVs and movies. And they're very popular and very iconic animal. But we didn't have those in Oregon. We had what was known as the Columbian mammoth, and they were in the Willamette Valley alongside the mastodons. And you can see from our little kind of cartoon rendering here that those are the dates that Danny ended up gathering from specimens within the Willamette Valley. And this means that those two animals basically were coinciding within the valley, living kind of shoulder to shoulder, as it were, even though they didn't really overlap. And this has everything to do with the environments that they would live in and what they ate. And that's the big thing that separates the two, is the habitat that they lived in. Your mammoth used to live in these open grasslands that was called a mammoth steppe, an environment that doesn't actually exist anymore today. Whereas the mastodon lived in woodland areas and riparian areas because they were browsers. They ate woody vegetation. And we can see that this big difference kind of split off about 30 million years ago, making them very distinct, basically from all the other elephants. And it helped them evolve to be quite different, even though when you just look at an elephant, they kind of look pretty similar. So the history of mastodons is really intertwined with the history of this country. This particular femur that I got to visit kind of pre-pandemic, when we could actually all go out and travel places, this sits in the Paris Natural History Museum. And this is a really special femur, especially in terms of paleontology, because it comes from a site in Kentucky. And it was basically one of the first evidence of mastodons that had ever been found. And it was located it was found and recovered in 1739, so very early, basically before the country was even established. basically setting the stage for it to be a very key component of early paleontological work. And it actually got wrapped into this larger argument and debate that was going on about the idea of extinction. This is around the time that the idea of extinction was kind of coming to its own, and mastodons were one of the key species that were used to basically prove that extinction was a thing and that animals didn't simply survive forever, that they did come into being and then sometimes were no more. And this is a very, very hot topic for President Thomas Jefferson, especially when he was in the White House. He was actually known to lay out mastodon bones within the White House itself, which is kind of a fun history fact. But he wasn't quite convinced of this extinction idea. And so there are rumors. It's kind of a story. It's not quite, we don't have any good documentation of it, but there are rumors that he actually told Lewis and Clark when they ventured across North America to keep an eye out for mastodons and other thought to be extinct animals, with the hope that they could basically demonstrate how unique and how important the New World was. Unfortunately, that wasn't the case. So just to give you an idea of how widespread mastodons were at one point, they have been found from Alaska to Honduras and have stretched over the entirety of North America. And one of the key things to see here is that we have a real dense concentration in the Great Lakes and in Florida. And this has everything to do with the habitat that they inhabited. These areas simply have more of that habitat, which allows a larger population, which usually creates more fossils. The other part of this is that there's also more people hunting for those animals in those areas because we have a higher number of population there in terms of human population. So the more people you have, the easier it is to find these things. And the more ground you dig up, which there's a lot of farming in these areas, there's a better chance that you're going to hit something. So that can kind of account for a little bit of the lack of dots, essentially, that we have in the West. But there's no doubt that there's quite a few yet to be found and yet to be recorded that have been found and simply just need to be put on the map. So let's get to kind of the heart of the talk tonight. This new mastodon that has been basically coined, this all came about from this group that's working out of California. This is led by Alton Dooley at the Western Science Center. And when we think about mastodons, we just kind of talked about how long they've been known about. It's almost 300 years. And so basically we kind of thought we knew what there was to know about mastodons. That there probably wasn't a ton to discover, and yet when they hit basically this kind of trove of mastodons at a large construction site in California, and they unearthed about 30 or so animals, they started to do some detailed measurements, and then they started to take those measurements and compare them with all of the other metrics and measurements that have been collected and published throughout the country and throughout North America. And they started to see an interesting pattern. The California mastodons seem to be a bit different than the rest of the country. And then they picked up on a few kind of outlier, interesting oddballs, a couple in Idaho, one in Montana, that fit this pattern too. And they started to kind of put all this together and realized, I think we have something new and special here. And they decided they had enough evidence to say that this is a new species. This is something completely different than other mastodons. And this is really a breakthrough type of moment because it's the first new Pleistocene proboscidean that's been identified in the last 90 years. It's pretty exciting stuff. So what are the characteristics of this new mastodon species? And basically, I kind of broke it down by the quality of evidence, with kind of the best being up top that we'll talk about first, and then some intriguing pieces of evidence that we think is pretty well supported. And then finally, some definitely some needs improvement kind of variables that need a little bit more research to figure out, and maybe some that should be excluded. So first of all, the best evidence we have is basically the shape and size of the third molar, which is basically the molar that's the furthest back in your jaw and in their jaws as well. And so to get an idea of what these look like, we have our American mastodon on the right there and our Pacific mastodon on the left. And you can see they're just a little bit longer, a little bit narrower, whereas the classic American mastodon is just kind of wider, more stockier. There's a little bit more talk about the number of lophs, which are those bands that you see. That's one of those equivocal variables that still needs to be looked at a little bit more. But this is the key one that really kind of defines the two species really well. And this is not uncommon. A lot of small characters can separate species pretty easily. All you have to do is have enough, basically, time and distance for populations to separate until you get these kind of unique and different characters that show us that they're different species, at least in terms of the paleontological record. So next, the ones that are the variables that are intriguing but probably need a little bit more work are that the femurs in the Pacificus, the Pacific mastodon, are shorter and wider. So they're basically more robust and more stocky in the Pacific mastodon. But there's so few samples. We'll talk about this a little bit later on, how the Tualatin mastodon fits into this. But it's an intriguing measure that we're still not quite sure if it's a good character yet or not. And then the other one is that in males, the tusks were found to have a smaller diameter at the basal end, which is the end that's near the skull. Basically, that it's smaller in the Pacific mastodon. And an example of this, just to get an idea, is that kind of empty hole in the tusk on the right hand side there, that area is basically smaller than it would be in an American mastodon. But again, this is another one of where sample sizes need to be increased a little bit more before we can say for sure that this is a good standby character. So the last ones are ones that are not exactly well supported. And this is because they're basically, both too few specimens. This is, again, a sample size problem, but it's also an issue where it's not well supported across all of the samples that we have. So this character is that the sacrum, which is your tailbone, which has a bunch of fused vertebrae in it. In Pacific mastodon, there are 6 fused vertebrae. Whereas in American mastodon, there's usually 5. Unfortunately, we know for sure in American mastodon that there is up to 4, or as many as 6 and as few as 4 have been recorded. And we just don't know how variable this particular measurement is. And so this was one of the least well accepted aspects of it. And the final one that was actually excluded from subsequent papers after the original paper came out is this idea of that the Pacific lacks any mandibular chin tusks. These mandibular tusks are just very small, kind of whisker-like tusks that stick right out of the chin, essentially, of these mastodons. And they're really neat little features, but we still don't know how or why they show up in the American mastodon, let alone whether they're certainly not present in the Pacific mastodon. So this is another one of those just kind of odd mysteries that keeps kind of driving us crazy, but we'll keep working on it. So on to the genetic aspect of it, of this recent evidence. There's been quite a few recent studies focusing on mammoths and bison recently. I know we're talking about mastodons, but there's been kind of this kind of wave, this push to look at all of these Pleistocene critters and their genetic variability and basically their genetic history. And the idea was, well, okay, we have bison and we have mammoths. We know that we know that there's different species, and we know there's different species of bison. So genetically, those should be supported. Well, it turns out they're not. Genetically, they're not different enough to be warranted as different species, which is really interesting. So even though we have an animal like a bison, where we have latifrons on that left side, which is just a gigantic bison, compared to our modern, basically tiny bison by comparison on the right side of that little grouping there. But you won't have the genetic variability within that group. So we really just think of those as what we call morphotypes. They're morphologically different, but not genetically different. So with that in mind, when we turn to mastodons, thought, oh, well, this is an animal that looks very similar. We assume is very similar. So genetically, it should be even less complex than either our mammoths or our bison, which have shown to be a lot less similar or a lot more similar than we had thought. But it turns out the mastodons ended up being much more different than anticipated. They were able to collect about 33 samples that had good results from basically across North America. And they tried quite a few spots, especially in Florida and a little bit in the Midwest, but they weren't able to quite get very good information, very good data back from it. But it shows that we have these distinct groups, what we call clades. And these are just we can think of them as families within the species themselves. These are just groups that are more related to each other than other groups. For instance, we have this group in Alaska and this group in the Great Lakes. The Alaskan group is this kind of pink-red down at the bottom there. And the Great Lakes one is clade G, this green grouping there. And we can see that they are related, but distantly. And geographically, they're very spread out. But then we also found quite a few oddball little clades that didn't make any sense and didn't kind of fit into this pattern. Which means you have a lot of change over time and basically a lot of population movements. And this has everything to do with the climate changes and with the glacials and how when the glaciers both moved down and expanded and then receded, this really impacted the mastodon populations because we found that they really don't like the cold. They're very temperature sensitive. And so unlike mammoths, who can tolerate that a little bit better, the mastodons would break apart and their little populations would continue to move. And so it would make sense for them to kind of stick closer together. And this also supports the idea that mastodons were similar to mammoths and modern elephants in that they were led by older females called matriarchs, and that we had matriarchal herds basically, where they would be the leaders of the group and kind of dictate where they would move around the landscape. But outside of those main groups, the ones in red and the ones in green, we had these oddballs that showed up, like the one in Nova Scotia, that purple dot, and the blue dot in Mexico, and then this kind of light blue dot that showed up in both Missouri and Alberta. And these are just kind of mysteries because when they're only represented by a handful of animals, we don't know how those populations kind of related to each other. And there's just not enough data to say for sure kind of how they all fit together. So that's one of our many ongoing questions, basically, which is, as you probably see, is kind of a theme of our talk here, that we have many questions. And so one of our big questions is, so if we have this new Pacific mastodon species, how do they relate genetically to these other groups that we didn't realize were so diverse? And how in what way do the genetics of these kind of Western United States mastodons kind of fit into the overall pattern? Could it give clues on what's going on with the other mastodons, or do we have something totally different that we're just completely missing? And so that was really the start of this project, was to kind of start filling in the gaps and try to better understand what is going on with mastodons out west. And luckily, in a combination of things, I was able to get in touch with Yvonne and come to find out that there is a creature called the Tualatin mastodon up there that was found was dug up in 1962 by John George and Ron Soon, although it was originally discovered by Marshall Charles Roberts in, I believe, 47. But this was just simply part of a college biology class. And they really did a good job in terms of excavation and recovery. You can see they were able to excavate the entire basically left side of the animal. And it was just in wonderful condition. And they documented it very well. And I believe they got an A on their paper as well. So that was a good job by them. And so what helped bring that animal to our attention was this paper and project by Danny Gilmore, which stemmed from his master's thesis project, where he dated several animals from the Willamette Valley. And one of them being the Tualatin mastodon, where he found that it had a relatively young date, about 13,300 or so, if we average our calendar dates. So fairly young. But very interesting is that it had a very good preservation record. And this is the part that we got very excited about because it has very good collagen preservation. That means there's a very good chance it has DNA preservation, which can help us basically start resolving some of these big questions we have about what's going on with mastodons, both out west and in North America in general. So, pandemic and all, I was allowed to come up to the Tualatin Heritage Center and do some basic recording to figure out if this mastodon was in fact one of these new Pacific mastodon species. So I went kind of thoroughly through and measured everything I could, especially your teeth, because the teeth and the molar is the most important for figuring out if it's a Pacific mastodon or not. And basically when I plotted it out and compared the known Pacific mastodon molars with the known American mastodon molars, the Tualatin mastodon fit very nicely into the Pacific mastodon, basically telling us for sure that we had a Pacific mastodon on our hands. It was a little on the small side, but we don't judge size. But the other thing to look at were the femur metrics. And this is where I was talking about the smaller sample size. There are really only 14 other femurs to compare to, both from the Pacific mastodon and the American mastodon. And we don't have a good handle on male versus female in terms of size and how they relate. It does appear that the data do separate pretty well, and they're statistically different. But again, your small sample size kind of undercuts whatever statistical answers you have. But on the bright side, Tualatin again fits very well within the expected range of a smaller female Pacific mastodon. So basically I think we have enough evidence to for sure say that our Tualatin mastodon is one of these new Pacific mastodons, which has a lot of interesting implications and ramifications. Because it expands the area. In a lot of ways, it makes sense because we have, you see a dot there in Idaho, a dot in Montana. It kind of makes sense that you can draw a big circle around this area. So Oregon, we always expected to probably have them. But the interesting thing is that you see those 3 dots up in Washington. Those represent 3 American mastodons. And those were measured specifically for the recent study that Dooley, the California group, did recently. So we know that those numbers are very good. We also know that at least one of those is a fairly late Pleistocene animal, which means these 2 populations, if they were pretty separate, lived in close proximity and remained different. And that raises a lot more questions in terms of how they may have interacted, if they did interact at all. And it kind of just opens more doors and basically requires us to answer more or address more questions and look for more data. But that's the nature of research and kind of the nature of science in general. So we're used to it. So when we knew we had good preservation and we knew going up there that ideally we would hopefully be able to sample the Tualatin mastodon, I knew we also had to do the ethical right thing, basically, which was record the tooth or record whatever specimen we were going to sample as absolutely thoroughly as possible. And one of the tools we can use for that is something called photogrammetry. And it basically just takes still photos. And when you take enough of them around the tooth or the object that you're interested in, you can basically take those photos and plug them into a computer program, and you get a very high resolution model. Basically, the more photos you take, the better your model is. And this has a lot of benefits, not only because of ethical reasons, that since you're sampling and you're doing destructive techniques to a tooth or to a specimen, you want to record that before you do any sort of damage. But it also is very powerful that you then can take this specimen and basically virtually send it to any other researcher or any other person around the globe. And it really brings, and makes the whole research world a lot smaller. And as much as we love to go to every museum and every location that has a specimen or something that we're interested in, unfortunately, it's just not practical. So this just provides another layer where we're able to then share and utilize these specimens in a more powerful way and ask new and interesting questions, but it also provides another way to have a permanent record of these specimens, so that worst case scenario, something terrible happens to the building and perhaps the specimen is lost or something. This provides another layer of backup, essentially. So luckily I did get permission to come up and to sample the tooth for DNA. And you can see it's a very, very small area that was sampled. It really just takes a very small piece of the bone, or in the root in this case, to get the DNA from the animal. And so this is just a short video where it's not very exciting because we're just taking a tiny little window out of this tooth, but it just kind of, it shows you how the process works. So we were able to sample it and we're able to ship it off, but unfortunately we're still waiting for results on that one. Unfortunately, we were also able to finally track down the piece of pelvis that Danny Gilmore had sampled for his thesis and that he published in that paper. It took a little bit of work, more than we had expected, but we did finally get it tracked down. And you can actually see where he sampled. That's that kind of blocky looking area on the right-hand side, or the left-hand side photo. And then we followed the same process to take a whole bunch of photographs and create a 3D model of the specimen. And this is a preliminary model, but it just gives you an idea of the high resolution nature of it. And this kind of opens the obvious question: why would you sample an animal twice? Well, there's a couple reasons for that. First of all, we wanted to double check that the pelvis actually did belong to the rest of the mastodon skeleton. Through various reasons, parts of the mastodon were separated. So we wanted to just double check and make sure that they were supposed to be together. But the other reason is more analytical, in that the more information you have in terms of the DNA, the longer and larger sequence you have. And the idea is that if, with the wonderful preservation here, this is our best shot that we know of to get the most complete DNA sequence from this new species. So when you sample in different parts of the animal, it allows you to kind of bring together all of the sequence together and provide a very complete sequence that we can compare to others, and to compare it to the American mastodon. And so since we can't talk about results of the DNA, I decided to throw this one in specifically for Mike Full, because he asked about the Tusk Metrics and how they fit in with the rest of the country, basically, and the rest of mastodons in general. And this is derived from a data set created by Smith and Fisher. And what they did was basically just measure the heck out of these mastodon tusks. And now these are all American mastodons from the Midwest. So we have a little bias there, and we don't know how Pacific mastodon really fits into that. But we do have a good separation shown by this basically maximum tusk circumference. So what is the maximum diameter or circumference around the tusk itself? And so while we have these two groups that are pretty clear, we do have this kind of group in the middle that were assigned as males based on other physical features within the skeletons, but are also kind of set out there in kind of a no man's land. So if you had just recorded those and found those without any other information, you might not know for sure which group to put those in because they just fall in the middle. And unfortunately, the Tualatin specimen falls right into that group. So we don't necessarily have a clear understanding just based on the tusks of exactly what the sex was. But this is just kind of in comparison to other mastodons in general. So on one hand, Mike is right. It is a little larger than expected. On the other hand, we don't really know what that means. And we're not sure how that kind of fits into all the other mastodons, and specifically Pacific mastodons. So kind of to wrap up here, the whole kind of thrust of this project was really to get our DNA samples from our Tualatin mastodon and figure out if it was one of these new Pacific mastodon animals. And based on our molar and femur measurements, I think we're pretty confident that it is a Pacific mastodon, which is pretty exciting because it expands the range, as we kind of talked about. It's very close to other populations. So now it basically gets the ball rolling in terms of new questions about where we can go next. And it also adds to this growing data set that is still being developed and still being better understood because it's still a little small, and we would like more to ask better questions. In terms of the tusk data, that it really starts to ask more questions. Because we don't know what the context is for how those tusks relate. We don't have a good record of males versus females and how they all relate to each other. And the simple fact is it could be an outlier. It could be just kind of a unique animal that is a little bit different. And unfortunately, in terms of mastodon data, we just simply don't have it yet. COVID has kind of made fools of us all in some ways. When I agreed to give this talk, I thought for sure we would have the results by now, but unfortunately, they're simply not in yet. But luckily, we have learned quite a bit and really, I think, made this project worthwhile. But that's all I have. So thank you very much. I really appreciate your time and coming out tonight.
Thank you very much, Andrew. Sure appreciate that. So we have some times for question and answer. Do we have someone who has a question? Okay, are mastodons found on other continents?
That's kind of an interesting question. Early mastodons, before we have American or Pacificus at all, some of the most, what we call basal, which means that they're the earliest in the evolutionary sequence. There's some debate on whether those show up in Asia, but not necessarily, that's not necessarily agreed upon. But doesn't mean they're just not there, but we simply haven't identified them yet. Usually what we see is things that aren't quite mastodons yet. They're basically pre-mastodons, if you want to think about it that way.
I had a question. Could you tell me, you mentioned that the mastodons on the East Coast, they were a matriarch. How do they determine their matriarch when they've never seen them or only seen the bones?
Very true. Basically, we look at patterns that we see in modern elephants, but then we also look to herd structures when we see large kill sites. So one of the really good ones are sites in New York and others on the East Coast where you have kind of large death assemblages, and those look very similar to those found in Africa and India with modern elephants. And we start kind of connecting the dots and do some kind of educated guesswork, basically, where it makes sense that we have older females in this mass death assemblage, just like we do in modern assemblages. So it's a bit of a leap, but behavior always is when you get into trying to get at bones, although I think it's one of the most interesting questions.
Okay, we have a question from Larry. Mastodons were browsers then like deer. Is that correct?
Yes. And just like deer, they were also usually browsers, but not always browsers. Whereas mammoths and animals with those very flat teeth, they only ate grass. Whereas mastodons were a lot more similar to bison, where they would just eat whatever they could. But primarily, they were browsers.
And then Paul says, does the age of the fossil allow extracting DNA that would not be possible from much older items, say like a T-Rex?
Yes. I don't know if you saw in the news recently, but they just found or just were able to recover the oldest known DNA from a mammoth that was around 1 million or just over 1 million years old. So basically, the older you get, the more degraded the collagen and the DNA will be. And so the more difficult it is to actually get to those specimens. And if you're asking about T-Rex DNA specifically, I have read a little bit about that, just kind of on a whim. And there is some debate on whether that it's actually T-Rex DNA or if that's a bacterial mold that would have been around and in the fossils themselves. It's a really cool debate, but it does show you that potentially, you could go very far back in time, but the simple rule is the younger it is, usually, the better preservation you're going to have, which also makes Tualatin a very excellent candidate.
I have a question.
Yes.
This is Yvonne Addington. This Tualatin mastodon has been a part of my life for almost 50 years now, since I was a teenager. And Portland State and Tualatin are probably very influential to you being able to still see the results of it. I noticed Scott Burns, professor, is on the line here, and he might have some questions too. But during your process, some people would say the mastodon we have was 29-year-old female. Then others would say, no, that's not right. It was a male. So now I'm asking you.
Well, and this was one thing I looked at, and I actually had a slide that I decided to take out at the last minute. But in terms of age, we can be a little bit more confident that it probably is around in that 30 or so year old range. That makes sense in terms of what the skeleton looks like. So basically, as you mature, your bones fuse together, the ends of your bones. And those fuse at very specific times, which helps us understand and identify how old an individual animal is. So we know for sure that all of the bones in the Tualatin mastodon are fused, but the teeth don't have a lot of wear on them. So it's kind of right in that kind of gray area of what age it would be. So I would say between anywhere from 30 to 40. I'm not exactly sure where that 29 or 27 years old age came from without looking at his notes. I'm just not quite sure. Dr. Wirtz's notes.
And Danny Gilmore and John George were going for their master's thesis at Portland State and provided a whole lot of the information. The other question that's come up just with you is, do we have 3 molars, or is that 1 molar with 3 parts?
You have 2 there. The one that I sampled was the 3rd one. So there's 2 that are connected that are on display. And then you can kind of see in the photos that there's another one somewhere. I'm not sure where that ended up, but there is another one from the animal. And you can see that from the photos and kind of subsequent analysis. So.
Thank you.
I got a question.
Yep.
In one of your charts, you indicated male and female. Among mastodons, did the females have tusks as well as the males, or were they just the males?
They did. They both had tusks.
They both had tusks.
The females in general just had slightly smaller tusks.
I see.
And that's something we definitely know in the American mastodon. But again, it's one of those things that we just don't quite have enough animals to say for sure how different they were between the sexes in Pacific mastodon. We believe that the pattern is probably going to be similar. That makes a lot of sense, but we'd like to confirm that with a few more specimens. So,
thank you.
Yep.
Okay. Would you surmise there are more Pacific mastodons located near where the Tualatin specimen was found?
It's absolutely possible. We clearly have an area that, along with the dike fossils that actually Yvonne was talking about kind of before we started here, which were found just right next door, basically, to the Tualatin mastodon. So we know this is an area that is rich in Pleistocene animals. How extensive those deposits or how many animals are there, we won't know until someone goes out and checks. And it's always the case with any science that involves excavation. But this is a good area and could have a very good chance of having more individuals.
Okay, why did mastodons and mammoths go extinct, but not in Africa?
That is a very debated question, a very hotly debated question. So I don't know if I will give you a final answer, but the general consensus is that animals in Africa basically were able to co-evolve with humans. And so they were basically able to kind of adapt and figure out how to live with each other. Some people think that when humans came to North America, that was a different situation where the animals weren't aware of humans, weren't accustomed to their hunting practices, and basically were killed off by them. However, there's better evidence that climate change was really impacting these populations, because we have quite a bit of data that shows that populations were declining all throughout the Pleistocene. And basically there's a steady kind of slow decline with the number of animals, number of dates, number of specimens that we see throughout basically the end of the Pleistocene, which all points to something happening before humans got there that were making the populations decline. And we really only have one site in all of North America where we have a clear human and mastodon interaction. And it's not a kill site. It's most likely some sort of processing site, or maybe they were simply scavenging or using their bones or meat or hide in some way. And so that's why we think that it probably wasn't really human caused, but at the same time, that question is one that plagues archaeologists a lot. And
I think Danny Gilmore determined there were no human marks or kill marks on this animal, and so he didn't think that it was killed by anything. The one that wasn't answered is if the Ice Age floods had anything to do with it.
That I can corroborate that there are no marks, there are no butchery. There's, as far as I see, humans had nothing to do, yes, with the Tualatin mastodon in any way. The floods are a more complicated question. If they did potentially wipe out a lot of these smaller populations, which is very possible, it could have had a huge impact on local populations, which basically could have what we call extirpation, which is them being made extinct in a small area. And so that could have had an impact that way. These are relatively slow moving animals, and those floods came through in a hurry, as we know. And even if they weren't killed outright by the flooding themselves, they modified, the floods modified the landscape in obvious ways, which would have impacted their vegetation, impacted how they would have lived and survived, and perhaps hurt them so much that the the local populations weren't able to survive. So that's kind of another one of those interesting questions. And so it's absolutely possible that the mastodon you have in Tualatin could be one of the last ones that was around, and it just didn't make it out. It was kind of clung on until the end. At the same time, we don't see a lot of evidence of stress, which we would expect if it was having a really hard time and hard life at the end, but not all of the stresses are necessarily recorded on the skeleton. So.
Thank you.
See now, I'm seeing if there's any other questions here. What about the Columbia mammoth? You didn't mention them. Are they the same as woolly mammoths?
So I did sneak them in right at the beginning. I was trying to focus on mastodons, but yes, we did have Columbian mammoths. We actually had more Columbian mammoths than we had mastodons. And they are related to the woolly mammoths, although as we found from recent genetic studies, they're not that dissimilar. They're actually very, very much the same critter, basically. They just look a little bit different and live in different habitats to some extent. But our Columbian mammoths actually outnumbered the mastodons in the valley for sure, and most likely in Oregon, because if you look at any of our kind of large databases of our fossils, there are a lot more mammoths than there are mastodons. And those are all Columbian mammoths.
All right. Let's see now, we asked that one. Thank you for an excellent update, is from one of our participants. Okay, are there any other questions that people have? So, Dr. Boehm, thank you so very, very much for your program. Really appreciate it. So let's give them a thumbs up or a hands.
Thank you all so much.
Oh, thank you, thank you. And as you get, if you have more research later on, please let us know. We would love to have you back again.
We will have information shortly on our genetics. So I hope to update you as soon as I can.
Wonderful. Thank you, thank you, thank you. So please join us next month for another program, and look for the update that will let you know our speaker and title. And thank you, everyone. We have another comment here from Scott Burns. Many thanks. Super job. So thank you, everyone, for joining us. See you next month. Good night.
Good night.
Thank you.